Today we're going to be talking about amino acids, peptides, and proteins. So I wanted to give you a quick overview about what each one of those terms mean. So amino acid, when we talk about amino acids, and I know many of you are familiar with the term amino acid, um, those contain a chiral carbon in the center. And this is a Fischer diagram, so it has some chemical implications, but I have COOH over here. So there's the acid part. On the other side, I have an NH2. So that's where it gets its amino acid name from. It has an amine group on the left and a carboxylic acid group on the right. And there's a hydrogen and an R group. And the R group is what makes the some 20 amino acids that are present in our body different. Now, if I take two amino acids and put them together, so I'll take NH2 and then C and then double bond O. So I'm going to draw this out. Now there's an R group here, okay, and a hydrogen. Then what happens is you form an amide bond. So if you remember from the carboxylic acids chapter, that's going to look like that. There's a lone pair on this nitrogen. And this is another amino acid, so I have a COOH on this end, like this, and another R group, another hydrogen. Now, this is actually known as a peptide bond. But anytime you get two or more amino acids bound together, that's what we know as a peptide or a polypeptide. A polypeptide would be many, many amino acids strung together. And then a dipeptide, which is what this is, would have two of them stuck together. So it follows the same terminology that we would for sugars. Now, when you take lots of these and you string them together and you create functional peptides that have a role in the body. Those are what we call proteins. And so proteins are really just much larger strands of one or two or maybe three or four. It can be many, many, many strands of amino acids that are put together in some sort of functional unit. So that's the idea. You have amino acids, peptides, and then if you scale up peptides, then you have like many of these, right? And you put them together in larger structures. This is going to lead eventually to proteins. Okay, so but I'll cover more of that later. So I just wanted to get you the idea of where we're going. And then I have whole sections on proteins and their structures and stuff like that. So first thing to cover that's maybe you've already thought about because I drew it as a Fischer diagram. There are D and L versions of amino acids. So D and L in sugars refers to the last chiral carbon on the chain that's pointed to the right or to the left. So for amino acids, we refer to D and L as in reference to where the amine group is. So this would be D, and then this is L. And it turns out that the L amino acids are the ones that are most commonly found in nature. As far as I know, we don't produce any D amino acids in our body. There are, in fact, 10 amino acids we can't actually produce enough of at all, but or produce at all for that matter. But the L is what we find typically in nature. Now it turns out one interesting amino acid that's a D amino acid is D-alanine, and D-alanine comes from bacterial sources. So we have a little bit of a D amino acid inside of us, but it comes from bacterial sources. So bacteria produce this in what's called a peptidoglycan. So basically it's a peptide bond with sugars, and so it makes this coating, and this coating helps to protect the bacteria from intact. So when we have D amino acids, it's often D-alanine, and it comes from bacterial sources. Now, bacitracin, if you're familiar with bacitracin, anybody who has children or gets scraped a lot probably is familiar with bacitracin. Bacitracin was actually derived from these peptidoglycans and contains D-alanine in its structure. Now, these are the 20 amino acids, and don't worry, you don't have to have them all memorized. I'm going to zoom in so you can see the whole thing. But it wouldn't hurt, actually, if I, when I post this, I'll also post up my lecture slides that I use occasionally. And probably the 4th or 5th slide in, there's this slide. And if you print it 6 slides to a page, it comes out the size of a credit card. And my students have often taken that and printed it out and laminated it. And then they just keep it in their wallet in case they have to remember what they are. Or their pocket, whatever they have in their pocket. And I don't know if they all have wallets, but yeah. So literally hundreds and hundreds of these things exist from students in past semesters that use these as references when they take their biochemistry classes. Okay, so anyways, I'll just point out there's some different kinds of ways that we organize amino acids. And one way is by whether or not they're polar or nonpolar, and so aliphatic means hydrocarbon side chains or hydrogen, and so there's a whole list of them here, and you'll notice that each one has a name. So let's focus on the aliphatic ones. Each one has a name: glycine, valine, isoleucine, I'll use my big mouse here, alanine, and leucine, and that also has a 3-letter abbreviation, which is gly, val, isoleucine, val, ile, I don't even know how you say that, actually, alanine, ala, and then leu. And oftentimes, when people refer to small peptides, they'll just say gly, val, and they'll just say the abbreviation. Technically, the name, though, if you wanted to put it together, if you had glycine and then valine bonded in that order, you would say the name would be glycine valine. And then they also have 1-letter abbreviations. And so there's G, V, I, A, and L. And those all make some very good sense. Now, let's look at some of the polar and acidic ones. Okay, so I have serine and threonine. And these are really alcohol side chains, and that makes them polar. That's why they fall in the polar subcategory. Relatively strong hydrogen bonders. Okay, and then when you look at the acidic ones, and these are the reason I'm really coming here, aspartate and glutamate. You notice the names look a little bit different, and this is actually also known as aspartic acid. Like that, and this is also known as glutamic acid. So you'll often hear both terms used, but they're really referring to the same compound. So, 8 goes to ick and ike goes to us. Those rules you learn from nomenclature, that's how you apply those to these guys. Now look at the letters for abbreviation. Serine and threonine, right, makes sense, but aspartate or aspartic acid and glutamic acid don't follow the same trend. So there's a nice little history, I put the reference up here, on how these amino acids got their 1-letter abbreviations. And you have to remember that there are 20 amino acids and only 26 letters in the alphabet. And back in the 50s, when they were beginning to use computers to analyze amino acid structures, one of the things that they had to do was come up with codes for each amino acid. And back then, 3 letters was a lot more than 1 letter, and so they coded 1-letter abbreviations. And this Professor Dayhoff, I believe, is who did this. Went about assigning 1-letter abbreviations, and some of the rationale for her abbreviations are in there. So it'll help you memorize these, should you have to memorize these one-letter abbreviations in the future. But for aspartate, it's aspartic acid, so that's how I got a D, even though it's not a D in the name, but she's just basically ran out of letters. Lysine is K, you'll notice that over there, because K is the next letter to L, and she had run out of letters at that point. So it's kind of, there's some of this is just kind of funny. And then glutamine got Q because you could say glutamine is the cute amine, like the Q, right? And so that's how that actually got its name. Anyways, at least that's how the folklore goes, and that's how it's written in that document. Yeah, so but histidine, proline, tryptophan, right? This is W, but for the most part, the letters make sense, and there's just a few that are a little bit odd because basically ran out of letters, ran out of the second letter, so they went to the last letter. Oh yeah, that's what I didn't explain about glutamate. Had run out of letters, and just so she assigned it the letter E because that's the last letter in the name instead of the first letter in the name. Anyways, there are different kinds of side chains, how we can classify them, and so we have heterocycles, right? And we have aromatics, but really these two are also aromatic, but these ones aren't heterocycles. So people will often classify aromatics and heterocycles, but those in fact both contain aromatic side chains. Now, I did mention earlier that there were 10 amino acids which we don't get enough of from our own body's synthesis, or don't produce them at all. Okay, and so these 10 have to be gotten from diet. So these are considered essential. This word essential means we get it from our diet. So we have to eat these. We'll say get via diet, like that. Okay, and so you know how people are. If they want to remember stuff, they try to rearrange them so it makes sense. And what they've done with these is they've organized them like this. Private Tim Hall doesn't represent, unfortunately, because this is the one-letter abbreviations, right? So it's kind of a mess. But represents the first letter of each letter of the three-letter or the regular the name of the amino acid. So P is phenylalanine, valine, tryptophan, threonine, isoleucine, methionine. That all spells private PVT, Tim Hall. The hydrophobics, should you have to memorize these? I just sort of threw them in sort of like one of those Scrabble calculators some time ago and figured out like what could I spell with these letters. And so the hydrophobic ones, I just came up with flag V imp. I don't know if that makes any sense. And then the polar ones with alcohol side chains or polar functional groups in them is cinch QT, and then LAH is actually an easy abbreviation for a lot of organic chemists because it's lithium aluminum hydride. Those are the basic ones, and the acidic ones are ED. So anyways, should you have to memorize them? For this class, you won't have to memorize them because, of course, this is online. So I would make these lists and organize them as you study for the test. So the next thing to talk about is some of the acid-base properties of amino acids, and really the reason we discuss this is a very important sort of physical property of amino acids that makes each of them different, known as the isoelectric point. So as you might expect, because you have an amine end, which can be a base, and an acid end, which can be an acid, at almost any solution pH, amino acids exist as charged compounds. Now, there is one pH for each amino acid when its net charge is actually 0. And so that's what the isoelectric point reflects. It reflects something what we call the Witter Ion, in which the amount of positively and negatively charged atoms in the structure are equal to each other and gives it a zero net charge. Okay, so before we can really talk about that, we need to sort of review how pH affects structure. Okay, and we talked about this. This was actually something discussed early on in chapter 1 or 2. And this is sort of a review, but I know at this point this is a whole different topic, and it can be a little bit confusing when we talk about it. So a Witter ion for an amino acid, and I've got one drawn down in the lower right of this slide right now, but a Witter ion would look like this. Got a negative charge, an R group, and a hydrogen, right? And then a positive charge like this. So NH2 is neutral and NH3 is positively charged. So that would be the Witter ion where I have a positive and negative charge. Now, at different pHs, let's say it's more acidic or more basic, it is possible that it's either just positive or just negative, but there really is one pH at which this will be completely electrically neutral. This point is also, when we say this is what we refer to as the isoelectric point. So I want to kind of explore what that means. Okay, so to start with, now let's talk about what pH means in reference to charge. And so I'm going to draw a line. This is going to be a number scale basically for pH. And let's say the pKa is, let's say it's 4. Okay. And so pKa, if you remember, is the equilibrium constant, which reflects how strong or weak an acid is. And lower values are stronger acids and higher values are weaker acids. Now, then we also have pH. So if the pH is below the pKa, so this is lower and this is higher. Okay, so my pKa of my acid is 4. That would be something like typically of a carboxylic acid. They're often around 4, like acetic acid's 4.7, I think, somewhere in that range. Anyways, the idea is this: if the pH is lower than the pKa, then what happens is you lose an H+. And if you're you gain an H+, and if you're above the pKa, if your pH of your solution is above the pKa, then you lose an H+. And that kind of makes sense because lower pH means more hydronium in solution, and so it's more likely that your acid will be protonated or have an H+ on it. But if the pH goes above goes to higher values, then that's lower hydronium, lower H+ concentration. So it's more likely that your acid will lose a hydrogen ion. Okay. Now, and you also have to remember that when we're talking about this kind of change in pH versus a pKa, that it's not an all or none kind of thing. So like if on this axis here, I have percent protonated, okay, then what happens is it's 100% up here protonated when it's really acidic, and then it drops down and goes to 0%. Oh, I didn't quite get the line in the right place. Watch, I'm going to cheat. This is going to bother me because I know it's going to bother some other people here too. It needs to be like right in the middle of this. That's where they're 50-50. So, in fact, right in the middle I have, when you're at pH equal to the pKa, it's half protonated and half deprotonated. If I go much lower, like 2 or 3 pH units, it's very close to 100%. And if I go much higher, it's very close to 0%. So the degree of protonation really depends on the exact pH, but in terms of general ideas, if I'm less acidic, or I should say at higher pKa's, less acidic than the acid is, right? Then what happens is it's going to become, let's say, 50% and then lower percentage of protonated. And if I'm more acidic than the pKa, then it's going to be 50% and then higher. Eventually it'll be nearly 100%. Okay. So let's talk about alanine. Alanine is one of the simpler amino acids. And I created the number line here. So in this number line, the carboxylic acid end has a pKa of 2.34. And then for the amine end, it has a pKa of 9.69. And so what we're going to do is analyze each one of these functional groups at these different pHs, okay? So to start with, let's just pretend we're at one end of the scale. Let's say we're all the way to the left side, so we're over in, sorry, use this pointer. I'm over in this region of the scale. If I'm in that region and the pH is low, right, then I'm below the pKa of the carboxylic acid, that's what this is, and I'm below the pKa of the ammonium ion, like this. Or of the, yeah, of this, NH3+. Because that's when we're talking about pKa for the amine, we're actually talking about the pKa for this guy. So below 9.69, the NH3 is protonated, and so is the carboxylic acid. They both gain an H+, or have the H+ on them. So what that means is overall, the amino acid is positively charged here. It's essentially +1 if you're at pH, like say pH 1 or pH 0 in that range. Now, on the other hand, if I'm all the way to this side, right, I'm all the way over here, then my pH is above both of my pKa values. So that means both of these will lose their protons. So the NH3 will just be neutral. It'll become NH2. All right, so instead of being NH3+, if I'm above 9.69, it's going to be just NH2. But the carboxylate group, right, I'm way above its pKa, so it's going to lose the proton. And as a result, it has a negative charge. So overall, this compound will be negative on this end. So there's a -1 and a +1. Now, somewhere in between here, and I'm going to simplify the somewhere in between here because that sounds very vague, but the somewhere in between here is when it goes from being negative to positive. In other words, it goes to being exactly 0, right? Because you can't go from negative to positive or positive to negative without being 0 at some point. Turns out that's exactly halfway between these two numbers, like right here. So this is going to be the Pi, or sometimes known as Pi or PHI, and I think they use a lowercase I for that. But in chemistry, for the most part, we just use Pi. I think PHI comes from biochemistry. All right, now, having said that, the calculation is pretty easy. That's actually what's shown down here. Right? Just average the pKa values, because if you average these two numbers together, you're going to get 6.02. That's exactly between the two of those values. So think about what's going on here. Like, remember I said 100% and 0%, right? So what's happening is this is deprotonating, and this is protonating as I go from low pH to high pH. And so what's going to end up happening at some point in the middle, there'll be an equal mix of both positive and negative groups on the molecule. So you form what's known as the Witter Ion. That's this guy. This guy lives right here in this range. This is where you find the Witter Ion. When the degree to which this amine group is protonated and the carboxylate group is deprotonated are exactly equal. Remember, it's degrees that we're talking about here when we're talking about pKa's. When the degree to which they're protonated and deprotonated is exactly equal, is right in the middle. And that's when, on average, right, the molecule is electrically neutral. And the calculation is pretty easy. You just take the pKa's and you average them together. But conceptually, that's how you put that together. Most of the amino acids can be treated this way. That is, you can calculate Pi just by averaging the two pKa values. Okay. Now, I'm just going to tell you how you do it if it's not the case, okay? And then I'll go through like why it's that way. So let's say you have an acidic side chain, okay? Or you have a basic side chain. So that means, for example, an acidic side chain would be a carboxylate group like this. That would be an acidic side chain. Now, I don't have a basic one here, so let me go back to this chart. Remember, we had acidic, and then we have basic, right? Lysine and arginine have these nitrogens that are here, like this, right? Or this. Those are basic side chains. Because the nitrogen's a base. Yeah, those are the only ones that really matter. And then there's a lot more acidic ones, or potentially acidic ones, than basic ones. Oh yeah, so where was I? Oh yeah, so how you do the calculation. This is how you do the calculation. If you have acidic side chains, right, you're going to average the pKa's of the acidic group. So the side chain pKa and the pKa of the carboxylate group in the amino acid, like parent chain, like in the long chain, that's the one you're going to, or the horizontal chain, that's the one you're going to be averaging together. So in other words, you'll average this one together with this one. That's how you'll get the PI. Okay. Now, if you have a basic side chain, right, then you average the pKa's of the basic groups. So let's go ahead and go through. Now, I don't have an example of basic one, unfortunately, but let's go through and show how you do this and then why you do this, right? So we're going to actually go ahead and do it, and then we're going to explain like why it's that way. And it requires a little number line and all that stuff. But to calculate it, I'm going to just do this. PI, it's 2.19 plus 4.25 divided by 2. I'll throw that in my handy dandy calculator. And that comes out to 3.22. So that's the PI for that particular compound. So let's go through the Y. The Y is actually a lot harder to understand. It's not impossible, but it's harder to understand than the how. Okay, so let's do the Y. So what I'm going to do is I'm going to draw this number line again, and we'll talk about what each functional group looks like at different pH ranges. So now instead of having 2 lines, I have 3 lines, and so the 3 lines, I'm just going to space them roughly proportionally like this. Okay, so it's not an exact number line. So this is 9.67. Okay, and this is 2.19. Oh, sorry, 4.25. My bad. And this is 2.19, like this. Okay. So now, if my pH is really high, so let's start there, okay, then everything will be deprotonated. In other words, the carboxylate groups will both be deprotonated, but the amine group will also be deprotonated. And so overall, I have a -2 charge up here. Now, as soon as I get below 9.67, right, then this will protonate. The amine group will protonate below 9.67. Again, let me point out to you, this is the one, right? This is NH2, and then, sorry, let me go like this. This might make more sense, actually. I'll do it like this. NH2 on this side, and then with a bond, and then NH3 plus on this side with a bond, okay? So I've protonated it, and the carboxylate groups are both deprotonated, and so the result is in the middle here somewhere, it's going to be -1. Okay? Now, so to think about this like in simple terms, let's go all the way to the left, right? So we're going all the way to the left side, very acidic. So if we're very acidic, both the carboxylate groups will be protonated because my pKa is lower than my pH is lower than my pKa. All right, so I'm at a pH lower than my pKa's. So that means down at this end, the molecule's +1. And again, between 2.19 and 4.25 is when I have to cross through 0. So my PI is calculated from these two values. So PI, right, is right. Let me just draw a little line like because I've kind of run out of space. That's where my PI is. It's right between those two. Because I've done the analysis and I know it goes from being positive to negative. It's in between those two values in which it crosses 0. So that's where PI has to be. And then again, that's why we're averaging these two values together. So let's take a look at cysteine. We'll do a couple other examples. And you really do have to think about these acidic and basic side chains. But primarily just the acidic ones are the ones where people generally have problems. If I'm looking at the acidic side chain for this guy, then what I'm going to do is I'm going to say, okay, I have NH3. I'm going to draw my line like this, right? And then I'll place the pKa values on the line just so I can think about it. It's not really quantitative, right? But it'll give you an idea. It gives you ideas of how to do it. So I have 1.71. I have 8.33 and 10.78. And what you have to remember about this one, this is why mentally it's tricky. That pKa is actually pretty high. The next one we're going to do is really high, and that actually does present some real problems. But that pKa is pretty high, so a lot of people misinterpret that as being a base. So you have to know that a thiol group acts as an acid, and when it deprotonates, would make, well, it becomes this actually, and then connected to the CH2. Okay, so you have to keep track of those kinds of things as you're doing this. You have to kind of know the chemistry of each of the functional groups. So at 10.78, again, I'm going to go through this whole analysis, and I like doing from high and then to low, and then I know when it's going to cross 0. Okay, so if I'm above 10.78, right, the pH is really high, then both these two groups will be deprotonated, so it's a 2- ion here. That means it's a 1- ion here. And on the far left, it'll be +1. Very acidic, it'll be +1 like that again. So again, 2 to -1, this one's +1, so it's between these two that you have to calculate the PI. The PI will be between these two groups. And again, the math is the same. It's the two acidic side chains because that's where it crosses 0. Okay, so then what I'll do is I'll calculate PI. 1.71 plus 8.33 divided by 2. That'll give me my PI. And that becomes 5.02. Okay, like that. So now look at tyrosine. Tyrosine is the one that really gives people real problems. And so again, I'm going to make my number line like this. And then I have 10.07. So that's the one that's the highest now. That's this. It's a phenol, essentially. And phenolic groups have higher pKa's. And then I have, let's see, 9.11, that's NH3. And then I have, let's see, sorry, I'm thinking about other things. 2.02 like this, 2.20 like that. Okay, so now, again, same analysis, right? Top end, very high pH. The very highest pH is 10.07. I go from being protonated to deprotonated. Like that. Again, the carboxylate end will be deprotonated already. That's all the way down here. And the amine will also be deprotonated. So it's actually -2 up here and -1. And then down here I'm +1, right? And so it's in between these two that I get pi. So this is where my pi is. This is pi. Now, why is that weird? Because when I'm doing that, I'm ignoring the side chain. And in all the other problems that I've showed you so far, you have to average the pKa's of the acidic groups or the basic groups. And for the instance of tyrosine, the pKa of the side chain is so high as it's not between the amine and the carboxylate in the sort of parent chain of the amino acid. And the result is, right, you calculate the pH as 2.20 plus 9.11 divided by 2. This is pi. And that value is 5.655, and then I'll round to 3/6 big, so 5.66, like that. Okay? All right, so again, you really have to think about, like, what does each functional group look like at the given pH, right? And then that allows you to calculate pi. So in a question, I might ask you, at which pH range is the ion -1 or -2 or 0, right? Or +1, and you'll have to know what each functional group looks like when you're at those pHs, and then you sort of add up their charges, and that tells you what the charge of the ion is. So one of the reasons we do this calculation of pi is it turns out pi is a method for the pi is used in a method called electrophoresis. So what I'll do first, before I talk about like how we do it for amino acids, I'm just going to talk about in general what electrophoresis is. So there's many forms of electrophoresis actually, but one sort of common way of doing it is to use what's called gel electrophoresis. And old school, it used to be called slab gel electrophoresis. And the way I'm going to describe it here probably is not very accurate, honestly, but it'll be good enough for our purposes. Basically, it's like a piece of Jello. It's a polyacrylamide. It's a polymer that has ions in it. Okay, so it's got a buffer in it to control the pH, and so the pH will become an important part of this, and it's going to be in reference to pi that we're thinking about what the pH is. Okay, so now we have a voltage, so we'll put a negative voltage electrode on this end and a positive electrode on this end. And in essence, what's going to happen is if you have a positive charge, right, positive charges will move away from so a positive ions will move towards the negative, and then negative ions will move towards the positive. So this will be positives like that. Let me redo that. So over here I have positive ions and negative ions. They're going to go in opposite directions. And the degree and the rate, so the rate of movement depends on charge. And the direction, the rate and I should say, and direction of movement depends on charge. So let's think about what will happen. Let's say I take, let's say I have 4 amino acids, right? You're trying to separate phenylalanine, methionine, tryptophan, isoleucine, and valine from each other. And you notice they all have PIs. Those are the PI values in parentheses. Between 5 and 6, basically, right? So what you'll do is you'll take a gel like this. So, and this is a top-down view. and you're going to place all of these amino acids in a single point. Use a pipette actually, and we put the amino acids in the middle like that. I'm going to put my negative voltage on this end, and I'll put positive voltage on this end. And this is what we do. We'll set the pH, okay, to be, let's say, I'm just going to pick a number in the middle, 5.74. Okay, so just randomly we'll set the pH equals 5.74. So what's going to happen, right? Well, so thinking about number lines again, because I always like doing it this way. This is the way it makes sense to me. Here's PI. If your pH is less than your PI, then that means you're more acidic, you'll have positive charge. And if you're above your PI, then you'll have a net negative charge. If you're at your PI, that's the only time you're exactly zero. So I put this voltage on, and I let the amino acids move according to their voltage in this sort of electrophoretic gel. So in the gel, okay, right? So now, phenylalanine at 5.48, his PI is below the pH, or I should say the pH is above the PI. So it's going to be negatively charged. So phenylalanine will end up, for example, like a spot over here somewhere. So over time, it's going to end up over here like this. So this would be PHE like that. On the other hand, the other amino acids will move in the other direction because they're all, the pH is below all of their PIs, if I set it up right. Yeah, they're all below the PI. So that means the tryptophan and the isoleucine and the valine will actually all move left. Okay, so that because they're going to be positively charged at that pH. I think I did that right. Yeah. Yeah. So then what ends up happening is according to their PI values, that's going to be in proportion to the distance that they move, because they're more charged the further away you get from their PI. So the one that's furthest away from 5.74, right, is the 5.96. So that'll be valine, would be over here. And then the next one is isoleucine. And then the next one is tryptophan. And then I set it up so methionine's pKa was actually the buffer's pKa. So methionine will stay in one place, and so it won't actually move. Now, side note, point number 4 down here. You don't need to know this reaction, but this is the Ninhydrin reaction. And Ninhydrin reacts with all the amino acids except for one of them, and makes a purple colored complex. And so the way you can actually see where these spots are, because you don't have no reason to know this, I suppose, but amino acids are colorless. So you wouldn't actually see anything. So the way you see this is you can use Ninhydrin to develop or make the spots colored so that you can see them. Now, having said that, right, there are other ways to do this technique. You can use this, doesn't have to be on a gel. It can be in a column. It can be in an open tube with solution, and there's other ways to detect them as well, because they do absorb in the UV light range. But just for seeing it by eye, right, by eye, then we usually use something like Ninhydrin to develop the plate so that we can see the spots. I should also point out this Ninhydrin reaction is very sensitive, and so like if you've ever seen a picture of a fingerprint that's purple, like you'll see these images like in spy shows or whatever, and they develop it. There's two reactions. See, one glows, but the other one's purple. The purple is actually Ninhydrin. What they're doing is as you sweat or you touch your sweat or moisture comes out of your skin, that contains amino acids, and so when you touch a surface, you leave this little print of amino acids, which can be developed using Ninhydrin. One other, there's actually two other ways that people commonly analyze amino acids. One's electrophoresis, one's thin layer chromatography. And in thin layer chromatography, you just take a piece of paper, in essence, a specially treated paper. Okay, and actually, let me draw what it looks like. You take a beaker, all right, and then you put the TLC plate on the inside, like this. You will have spotted your material down at the bottom, like this, and then you put liquid in here. And then what happens is, as the liquid moves up the paper, or the TLC plate is what we call it, so TLC plate. And the fancier ones come on a plastic or glass backing, and they have a material on it that's not paper. But these spots will move according to their polarity. If you took Chem 1A at Fresno City College or Clovis, or I know none of you maybe were in my 1A class, we do TLC of ink colors, and that's actually another way you could do it. And the third way that it can be done is known as ion chromatography. And ion chromatography is also a very good technique for to determine the amino acids in a mixture. I'm not going to spend time talking about it. There's a long description in your book, but I just wanted you to know like another way is ion chromatography. I can't spell chromatography in the bounds of my window here. Chromatography. That's weird. Let me just rewrite it. I tell you. Ion chromatography. Such a long word. There we go. Barely got it in. And what they do is they take a column and they put the amino acid mixture at the top of this column. The column is made up of like well, it's really silica gel or alumina or other types of material. Those are really like sand. And then they run a buffer through it, and based again by pH, the ions will separate. Remember, all the amino acids at some pH or the other will be in ionic form, except the ones that are neutral. And what will happen is as that buffer goes through the column, depending on how strongly the amino acid adheres to the column, it will separate out. They'll move at different rates, and then they separate from each other. And then we can collect the liquid that comes out and analyze it to see what amino acids that are in there. So I'm going to talk briefly about the synthesis or the making of polypeptides. And really, if you think about what a polypeptide is, or a peptide is, it's this amide bond that's in between here. Okay, so that's what we're trying to make. But the problem is at physiological pH, I have a negative charge here. And remember, what's going to happen is the nitrogen is going to have to be a nucleophile to this carboxylic acid functional group. And so the result is there's not a lot of ways that you can do this with the functional groups without modifying them, because the nitrogen will be protonated, and so the lone pair that it would use to attack as a nucleophile is going to be blocked by being bonded to something else. And then the carbonyl carbon, this carbon that's right here, okay, won't have as strong a positive charge, because the negative charge that's on the oxygen helps offset some of that. So it's not very electrophilic, and it's not nucleophilic, and so you can't really make the reaction take place. So what people do, the strategy is to activate one end of the amino acid by replacing the carboxylate hydroxy group and replacing it with a better leaving group. But here's the problem. I mean, that would make total sense. Let's say you could make this into a chloride like this, right? And let's say you could run it at pHs where this was not protonated. Well, then this could be a nucleophilic reaction on an acyl halide, and that would be a very fast and easy reaction to do. But sort of the basic problem, though, beyond that, making those modifications, is that if you do activate one end, what's to keep it from reacting with a molecule of its own? That is, what's to keep it from bonding to itself over and over again? And really the answer is there isn't anything to keep it from doing that. So the strategy is this, okay? In order to make this bond, you have to actually protect this end, and then you have to activate this and make it into a good leaving group. And then once you do that, then you can add your second amino acid in. And that amino acid come in and add and then create the peptide bond that we see here. Okay. So another strategy, though, and I'll just mention this, okay, is to actually protect this end, right, or block this end, and then just bring in an activated amino acid, and then have it react with the free amine end. So both of these strategies actually get used. That is, the way that we build amino acids. The way I mentioned first, since I'm adding to this functional group the new amino acid, this is known as an N to C. If you go from the other direction, it's C to N direction. It's going backwards, okay? That's the way I think of it, is always backwards. Okay, as far as naming goes, this is kind of miscellaneous information. That's how you would name it. You just kind of jam all the names together and end them in YL. So cystenyl, methional, cystenyl aspartate. And then, yeah, anyways, it's kind of a mess. You don't usually see people doing that. Okay, let's see. And then I have some other notes written in here. So the strategy is to do this again, is to protect. So I have a carboxylic acid, I have an amino acid. So I'm just to draw it like this, and I'm not going to put groups on there, but I have an NH2, and then a COOH like that. Okay, I'm going to zoom in so you can see it. And then I'll take and I'll protect this group. Now by protect, I actually mean react it with something that keeps it from reacting when you don't want it to. And then we'll activate this group. So then what you do is you bring in an unprotected amino acid. And then you form the amide bond here. And so what you'll end up with is you'll end up with something that looks like this. Let me draw the structure first. NH2, and then C double bond O, and then this is the peptide bond. It looks like that. And then this end is protected still. So then what you do is you activate it again. So I'm going to cheat here. I'm just going to copy this over. And then we activate this end again. Oops, sorry, I need to, I don't know why it didn't write, but let me get the pen back. I activate this group. Oh, I used a box for that. And then you bring in another amino acid. So that's sort of the process, okay? So that's N to C. Now, if you go from C to N, this is something that a guy named Muirfield did. And You can read about it in the book. It is an important technique. But what he did was he took basically a polymer, okay, and so I'm going to draw the like beads. Okay, these are beads of a polymer, and he bound the C terminal. So oops, that's a single bond of a carboxylate of the carboxylic acid to the polymer like that. Okay, so this is Muirfield, and I'll write his name here in a second. And then what he did was he brought in a protected activated amino acid like this. So I have the protection on this end, and I have the activation on this end. So he brought it in, and then this would react, and so then in the next step, in the next iteration of this, on these beads, right? You would have a dipeptide. So that would become this. Now, this end is still protected. So what he would do is deprotect that end, okay, and then add, and then repeat the sequence and add this protected activated amino acid in, and then you'd go from a dipeptide to a tripeptide. And here's the reason why he did this. He could automate this process, all right, and he could use purified reagents at each step. Think about what happens when you try to do this method, okay, this method N to C. Every time you make an addition, you're relying on protecting one end, activating one end, adding an amino acid, activating one end, adding an amino acid. And that process turns out to be not 100% efficient. And so if you want to grow an amino acid peptide, an amino acid chain to about 10 amino acids, that would be like an oligopeptide, right? That's the term we use. If you want to make an oligopeptide, it becomes very difficult to maintain fidelity over the course of all those reactions. So let's say 1 in 10 doesn't work, right? So then the first time you do it, you have 90% of your product and the other 10% is something else. And then you add the next amino acid and then 90% of your product, right, and 10% is something else. But this process is multiplicative. So eventually, if you have a 90% accuracy in adding amino acids, and it's actually probably higher than that, but if you have 90% accuracy in adding amino acids, the way you calculate your accuracy, let's say after 10 tries, is to take 0.90 to the 10th power. And by that point, you've only got about 34% of your product that you actually want, and 66% roughly is something else. And so your yields go way down after you try to do many, many, many iterations of this thing. Now, the advantage of the Muirfield method it might be one R in there, I don't quite remember, but Muirfield is that because the amino acid was bound to this column, he was able to wash it out and purify it at each step. So he would bond the first amino acid, and he would wash out all the impurities. He would add the next amino acid in its activated but purified form, and add that till it completely reacted. Then he'd wash off all the unwanted material, and then he would bring in the next activated, he would unprotect the nitrogen end and then bring in the next activated protected amino acid. And so he could very carefully control this and get much, much higher percent yields, and was able to actually, I think he was able to go up to about 100 amino acids this way with reasonable yields. Now, these are actually the reactions, and you don't need to know them, but I thought I would put them on a page in case you take a biochemistry class. You need to know them later. I'm not going to really describe them, but those are actually, the protection group is TBOC, okay, and it adds this to your amino acid. The nitrogen is part of the amino acid, but it adds that group on there, and that keeps that nitrogen from reacting because it's part of an amide type bond. And then DCC is the activation. This is actually the leaving group right here. Okay, so that's your leaving group. This is your protected end. So that you can see why I didn't draw those earlier. They just make things very confusing. But if you've worked with this at all, if you take the higher level of organic chemistry, for example, and you work with these structures don't become that intimidating. So this next section is usually titled something like determine protein structure, but really we're not determining the protein structure. I'll go over how you determine a primary structure, but principally what we're going to be looking at is what the different levels of protein structure are, and then we'll talk about how we determine primary structure. So primary structure by itself is the sequence this doesn't belong here actually, is the sequence of amino acids. So, alanine, glycine, methionine, whatever the order is, that's the primary structure. Now, the secondary structure contains alpha helices, beta pleated sheets, and then this is also disulfide down here. That's where that actually went. Disulfide bridges actually come from cysteine, the amino acid cysteine. So if I'm drawing the amino acid cysteine, looks like this. There's a CH2 and then there's an SH. It turns out when you take two cysteines in a structure, and let's say my primary structure is a long strand and each one of the little dots could be amino acids, for example. I get a long strand like this. And it'll actually have structures built within it that are called alpha helices and beta pleated sheets. And I'll show you those in a bit. But these disulfides, I can get a sulfur bonded to a sulfur on another part of the structure, and that actually keeps that part of the molecule folded up. So an alpha helix is a structure that looks like a little coil, and I'm actually going to show you like simulations of these. They're actually not simulations; they're actually modeled structures, or actually structures determined from different enzymes and proteins. And then you'll have these long sections like this, and so you'll have helices. They're literally coils. This would be an alpha helix. And these are beta pleated sheets. And so when I drew this up here, I was sort of generalizing just strands, but really what the secondary structures look like are the alpha helices and beta pleated sheets. And then in the secondary structure, you might also have disulfide bridges, but really that sort of comes in with tertiary structures when I'm talking about disulfide bridges, I guess. And so I guess I didn't think too clearly about that, to be honest. That really should maybe even be down here. But those that secondary structures are alpha helices, beta pleated sheets, includes some of the disulfide bridges, but the tertiary structure is how all these larger units, like the alpha helix and beta pleated sheets, actually interact with each other and fold, is what they call it, fold into a sort of a larger unit. So So I have primary sequence, secondary alpha helix, beta pleated seat, tertiary, how those units interact with each other. And then we have quaternary structures, and quaternary structures are actually multiple tertiary structures aggregated together to make up a functional protein. And the protein functions, there's all kinds. There's like transport, there's structure, there's enzymatic proteins that facilitate reactions. And I'm not going to get into all that long list because that's really something you'll learn in biochemistry. But the quaternary structure is what we think of as the end product, like the machinery of the cell. So now what I want to do is show you using a program what these structures actually look like. So this is an alpha helix, and I like using this program. And for some of these, I'm going to have to use a computer-based program. But this is a little program that you can get and you put on your computer and just run it from your desktop. But this isn't actually an alpha helix. I don't remember what structure it's from, but what actually holds the alpha helix together and gives it its shape is this. Actually, let me go down in here. Let me see if I can find one. Yeah, we can recognize this. This is valine right here. The red is oxygen, right? And the blue is nitrogen. Yellow is sulfur. So this looks like it's probably methionine. Yeah, most likely. And then, so anyways, sorry, get back on topic. What holds this together and gives its shape are two things. One is hydrogen bonding, okay? Hydrogen bonding is going to happen between the hydrogen on the nitrogen here, so it's sticking out like this, and then the oxygen on a carbonyl of another peptide bond. Okay, so this hydrogen is a nitrogen on the this hydrogen is on a nitrogen, and that hydrogen is hydrogen bonding to the oxygen on another peptide bond. Now, the other thing that determines this shape is the groups that are on the outside. And you'll notice typically what happens is if you have large groups, they tend to form these alpha helices because sterically, if you wrap the peptide chain like this, you can put the large groups to the outside and they get more space. So hydrogen bonding and then steric considerations are really what the molecule is doing in order to make the structure. Now when the groups are smaller, often it'll make a beta pleated sheet. It doesn't have to be smaller, but oftentimes when it's smaller, you get beta pleated sheets. Now, this is just one way of looking at the molecule. The other way of looking at the molecule is to follow the peptide bonds. And remember, when I drew the peptides, I drew them like this. Well, I didn't draw them like this exactly, but this is what they look like. And I have a lone pair and a hydrogen here. That's the hydrogen bonding. Now there's a resonance structure that goes like this, because nitrogen is a very good electron donor, and you get this sort of resonance contributor that looks like that. Negative, positive, like that. And so what happens is, because this bond has a lot of double bond character to it because of this resonance contributor, this part of the molecule stays relatively flat. So going back to this structure, you'll notice that in the regions of the peptide bonds, it's relatively flat. So let me zoom in again. So remember, this is a peptide bond, right? This region here. It's relatively flat. Relatively flat. That's again here. Another one here, relatively flat. And normally, right, if it's double bonded, it's going to be completely flat. So this is giving us the helix's sort of turn radius. So another way to look at this, though, is to look and follow that flat ribbon that goes around like this. And the style is actually called ribbon. We're going to go to ribbon. And it makes it really, oh, whoa, I don't know what happened to my ribbon. It disappeared. So there we got it. Here's the alpha helix in ribbon form. And then what I'm going to do is I'm going to choose the color options. so that you can see each of the amino acids in a different color. And I don't remember what the code for this color scheme is, but it's called Shapely anyways. And you can see, like when you look down the alpha helix, you can see that it's hollow. And you have to remember, if we were to put the groups on here, like we were to combine two views, that the larger groups would be on the outside. And that's part of what creates, or is the driving force for creating the alpha helix. It's the hydrogen bonding along the inside between the peptide bonds, and it's the large groups on the outside. So that's alpha helix. Again, and the primary structure, just put all this back in reference, is just a sequence of different colored amino acids. It doesn't have anything to do with the actual large scale shape. So let's look at a beta pleated sheet next. So here's a beta pleated sheet, or a section of beta pleated sheet. I don't know what the protein structure is. I believe this was a simulation. And when a beta pleated sheet forms, what you can do is if you display it as ribbons, and this is again the easiest way to see this, you can see this region where it's side by side. Okay, so it's not helical, but it's side by side. And then this is one strand going in one direction, and then another strand going in the other direction. So recognize when this strand is long enough, right? This piece could actually come around and then go in this direction. And I'll show you some of those, but you can have both parallel beta pleated sheets. This one is not parallel. It's called anti-parallel because they're going in the opposite direction. Like this is N to C and then C to N. So they're actually going in opposite directions, even though it's the same strand. If it came back around, then it would be parallel. Right, so you'd have a beta pleated sheet, some random segment, maybe an alpha helix up here, and then another strand coming in. They would both be going N to C direction. So now what I'd like to do is show you how these things interact with each other to form the tertiary structure. Okay, so I'm going to pop up a tertiary structure, and we'll look to see if we can find the beta pleated sheets and the alpha helices. Okay, so I pulled this one up. This is actually a real enzyme. This is an HIV protease enzyme. And this is the enzyme the HIV virus uses to break down, I think, genetic material. But anyways, looking at it like this, right, it's really hard to see any sort of secondary units, right? You might be able to see some. So what I'm going to do is I'm going to make this into the shape, into ribbons, and so we can follow the backbone, and then we can see a little bit more of its structure that way. Right, and then right there you can see these parallel strands. I think these might be parallel strands, and some up here. These are making beta pleated sheets. Now they're not maybe as clean as some of the others, but they're there. And then you have an alpha helix here. Now I'll put it in the shapely. I like doing that because it helps give it some more contrast. Right? And so now you can zoom in on the alpha helix like this, and you can see the alpha helix, make it stand out a little bit by turning it. And then you can see these beta pleated sheets here. And if you really wanted to know whether or not it's parallel or anti-parallel, you would have to like sort of follow the strand. This one here, though, this one's easy to see. You can see it's going in one direction and it loops back along itself. So this is similar to the one I showed you before. It's kind of what you see in silk as well. And maybe I should show you that one as well because that one's interesting. It's just very long repeating patterns. Anyways, this is the idea of a tertiary structure. The secondary structures interacting with each other. It doesn't show disulfide bridges in here, I don't think. So we're not seeing any of that. If you go online, you can actually use the web browser version. I think it'll pull some of that up for you. All right, so let's go back. And so this is tertiary structure. And now what we're going to do is look at a quaternary structure. And this is going to be a large molecule, so when you see it, you'll realize how big these things can actually get. So this is a ribonuclease or no, it's Rubisco. And what Rubisco does is in the Calvin/Krebs cycle, if I remember right, it adds the CO2 to the molecule. So this whole thing that you're looking at here is used to incorporate a CO2 into a sugar or into a precursor for a sugar. So let's take a look here. All right. If I turn this, you can see, like, I'm going to zoom in here and just look at the edge. There's an amino acid. So this thing is huge. I think it's like 10,000, 100,000 something. It's a big. Anyways, it actually consists of 12 subunits. So 12 secondary structures come together to make this one structure. So let's look at some of the options here. I'm going to do, where'd it go? I'm going to do, ribbons, like that. And now you can see all those secondary unit structures. You can see lots of alpha helices in there as I turn this. And then you can see some beta pleated sheets. They don't stick out as well, but you can see a bunch of them over here. All right. There's a few others I know. There's some right in this area here. That's not the same one, right? It's different. Yeah, and then we can put some different kind of color schemes on here. So here what I've done is the separate chains have each got a different color. So now you can see the subunits and how tightly interacting they can be, right? And I think, if I'm not mistaken, there's 12 different ones in here. So like this red one would be one, and this sort of tan one you see is a different one, and I believe it's still connected. So it's actually holding the blue, the red, the blue, and the other color. I don't even know what that you call that, dark brownish, together. Yeah, so you can see then individual strands, and you can also do individual amino acids, but in this scale, like if I do this, it's impossible to see anything, okay? So, yeah, if we look at the individual chains, then we can see all the different subunits. And so this is a functional protein, or close to a functional protein at least. I think this is one actually where they, and I don't see where it is, where the substrate of the enzyme is trapped in here somewhere, but I don't see where it is. Yeah, I don't see it. I think it's supposed to be in this image. So anyways, a lot of times what they'll do when they take images, this is actually from a technique called X-ray crystallography, and so they know the relative positions of every atom in the structure, and what they do is then reconstruct this image based on the spatial data they get for every atom. And then a lot of times to figure out where the functional portion of the molecule is, so this enzyme's giant molecule, right, or the enzyme is they'll actually take the substrate, the molecule that it works on, and they'll crystallize it with the substrate in place and without the substrate in place. And what that allows them to do is look at the shape of the enzyme as it changes when it binds onto the substrate, and it also gives them the information about where the substrate binds and how it binds into the enzyme's pocket, right? That's the key, basically. So again, different units of structural organization. There's primary, which is sequence. There's secondary, which is alpha helices and beta pleated sheets. There's tertiary. Each one of these different colors is a tertiary structure. And then there's the quaternary structure, in which those subunits come together and make a functional protein. So enzymes, things like that. So the last thing to do in this chapter, which is almost just like problem puzzle solving, is to look at how the methodology that's used for figuring out what a primary sequence is. I've put a couple of notes here together, but really the way people learn this is best is by actually doing it. And so let me explain to you how it's done. Okay, first of all, you have to cleave all the disulfide bridges, and so they usually use a reagent to do that. And then and the iodoacetic acid is the one that's used to do that, and so you end up having these acetate groups attached to the sulfide bridges. And then so once that's done, once you've capped off the disulfide bridges. So basically, you have a sulfur from a cysteine bonded to another sulfur from another cysteine, like this. But what ends up happening is this gets broken, and then you end up, well, you cleave it, and then you cap it, they say, with this guy. That's iodoacetic acid. And once you've split this, hydrolyzed it, then they actually naturally like, apparently, to bond back together. You cap it this way, and then once you have that acetate group on there, apparently you can't reform the disulfide bridges. So we break up the disulfide bridges, and then we cap them off, and then what we do is we hydrolyze the peptide, so the amino acid sequence, to figure out what amino acids are present. Okay, so once you know the amino acids that are present, we use something called Edmonds Reagent. And an Edmonds Reagent, what that does is it removes the N-terminal amino acid. So this reagent called phenyl isothiocyanate is PITC, ends up cleaving off the last terminal amino acid, and then they identify that amino acid. Now, when Edmond did this method, think about it like this. You have a protein, and you're cleaving off the N-terminal, right? And then you cleave off the N-terminal. You can actually repeat this process quite a few times, but what ends up happening is it's not always 100% successful. So maybe the first time you do it, you only get one amino acid residue. It's a PTH amino acid residue. You get one residue and you can identify it. And you can keep doing that, but occasionally you're going to have one that doesn't come off. So all of a sudden you have impurities, and these impurities build up over the sequence of cleaving off the end amino acid. So it's only useful for sort of a smaller type of protein. I think I've read that you could do up to 50 this way, but I think it's pretty unreliable up to 50. Okay, but I don't know that for sure. But anyways, it has limitations in doing that method. So another I guess technique that you have to be aware of is you can also determine what the C terminal is. So Edmonds reagent tells you what the N terminal is, and then you can use what's called the exonuclease. An exonuclease cleaves off one of the ends of the amino acid. So carboxypeptidase cleaves off the C terminal end. So Edmonds reagent can tell you what's on the N terminal, and carboxypeptidase can tell you what's on the C terminal. Now there's two of them, and you don't really need to care what it is. There's A and B. You just need to know that when you do that technique, you're going to get the C terminal's identity. Okay, so you have a protein sequence. It consists of amino acids. So I think I drew 8 here. So that would be an octopeptide. Edmonds reagent will tell you what this is. And then carboxypeptidase will tell you what that amino acid is. And why that's important is because the way we're going to do this sequencing, unless you know one end or the other, sometimes what happens is you can develop a sequence that could be from either C to N or N to C. So in other words, the sequence itself doesn't necessarily give you the order, the direction in which they're connected. It just tells you the order they're connected, not whether it's on the C terminal or the N terminal. So knowing which one's on the C terminal and which one's on the N terminal gives you a much higher probability of being able to give directionality to the sequence. Okay. Now, the next thing that you do is you use a partial hydrolysis to break up the peptide, okay, into smaller fragments, and then we analyze those fragments. So The types of reagents that we use are listed in this table, and you'll have access to this, of course, when you take your test. But this tells you where each one of these acts. So, for example, I talked about Edmonds reagent, takes the N terminal off. Cyanogen Bromide, though, takes and hydrolyzes on the C side of methionine. So let's say I have Alanine, glycine, methionine, and leucine, like this. Okay, and I use cyanogen bromide. The C side would be this side, and so I'll end up with two fragments. This one will be one of the fragments, and the other fragment will be the single amino acid leucine. So in fact, if you did this technique and you have a methionine in the first fragment and a leucine by itself, you know the leucine is actually also the C terminal of the amino acid. Okay, and then there's the endopeptidase. So an exopeptidase, right? That's carboxypeptidase A and B. That tells you the C terminal. But then the endopeptidases cleave at specific sequences within the peptide. So for example, trypsin is always on the C side of arginine and lysine. Okay, and then chymotrypsin is always on the C side of amino acids with six-membered aromatic rings: phenolphthalein, tyrosine, and tryptophan. And then elastase is on the C side of the small amino acids. There's a glycine and alanine. So we use these tools to partially hydrolyze or cleave, so chop up, in known ways, the peptide. And then we can use that information to generate a sequence for the peptide. Okay, so now, having said all that, I'll show you how it's done. So here's our first problem. And we'll work through this problem. And then, just so you know, I always try to keep this chart handy somewhere. Now, for us, it's probably easier on this problem to go to the top. I'm going to do examples 1, and maybe I'll do example 2 as well, but then I'm going to leave example 3 for you. That's this guy here, okay? And you see if you can figure that one out. But almost always, one of these ends up on your exam. So let's go ahead and get started. So I noticed while I was doing this that I missed one of the amino acids in this list. So I'm going to add it in here, but phenylalanine should be in this list. And this list is alphabetical. So phenylalanine would be like right here, PHE, like here. Okay. Now, this is what the data looks like. You're given a list of the amino acids that are present, and then you're given some chemical reagent data and endopeptidase data that shows different fragments that are formed. So the first thing that you want to do in these problems is just count the number of amino acids. And when you count this, what you do is you end up getting 8. So I draw 8 lines. So this is known as an octopeptide, right? So 1, 2, 3. Maybe make them a little bigger because I got to write in those slots. 1, 2, 3, 4, 5, 6, 7, 8. I'm going to go ahead and number them 1 through 8. And then I look at the data that's given to me, and I refer to this. Oh gosh. Fortunately, this program lets me drag the little window. It doesn't lock the position of the window. Let me see if I can move this back to where it was. It's an awkward position on the screen. There we go. I really want it up here. I'll just put it up here. All right. So I go back to this chart and I look at the reagents that are given, right? And I can see like Edmonds reagent and carboxypeptidase, and then I can look at this data and it makes sense to me. Now carboxypeptidase cuts off the C terminal amino acid. So I already know this is serine. Okay, so I fill that in. And Edmonds reagent gives me leucine. It gives me the first amino acid. So now what I have to do is I have to place the remaining 6 amino acids in slots 2 through 7. And we're going to use that using the rest of the data. So for cyanogen bromide's role is to cut on the C side of methionine, right? Now, you'll notice leucine is here. This is a pentapeptide. There's 5 amino acids in there. Methionine must be the last one in that sequence. So methionine must be number 5. Now, the remaining amino acids, right? So I've taken care of the leucine and the methionine. The remaining, the arginine, the lysine, and the tyrosine go in between. But we don't know the order yet. Now, the reason I say methionine's in slot 5, to be really clear, right? When you cut on the C side of methionine, you're getting a fragment that's 3 amino acids long, and the methionine is part of a fragment that's 5 amino acids long. So it has to be in slot 5. Okay. Now, for slots 6 and 7, I know serine's the last one, but in the middle, I could have arginine and phenylalanine. Now, I don't know the order. This is just alphabetical order when you're given these lists. So now what we have to do is we have to use the data from trypsin to try to sort all this out. So again, it's super important that you know what each one of these these endo or exopeptidases do, or chemical agents. And so you go back up to here, you see trypsin's on the C side of arginine and lysine. Okay, so that's right here. So I'm going to put this, arg and lys. These are the things I'm looking for. And so I have a lysine here, and I have arginine here, and I have an arginine here like that. So those are the ones I'm spotting. Okay, I'm taking a look at those guys. Now, I know leucine is only in once here, right? So I have leucine, and then I have lysine and tyrosine. So that means these two have to be next to leucine. So actually arginine and these lysine and tyrosine are not in the order that I've written. You have to reverse that order. So I'm going to write this down here. I'm going to write these slots again. May not have left quite enough room for this, but I'll try it. So I've already filled in this. I've already filled in this. I've already filled in this. And then underneath here, I can have lysine and tyrosine. And then I can have arginine. Make sure I did that right. Lysine, tyrosine, and arginine. Yeah. So arginine actually has to be in this spot here. That's the only spot left for it. So I can sort of fill that in. I'm going to do that in red. Then the question is it lysine, tyrosine, or tyrosine and lysine? And the important information that you get is that arginine is by itself, which means it's cleaving everything to the right. Like this is cleaving right here. But the fact that arginine is by itself means that lysine has to be next to it so that it ends up by itself. So then that means this is lysine, and that this is tyrosine. So that's the order for the first half. Now, some people will actually do this and get the second half and then come back and do the first half. The actual order doesn't matter in how you do this. So let's look at the last piece of data. I have serine by itself. So that's a really important clue, right? Arginine and lysine, I have an arginine and a phenylalanine, but the arginine has to be here because that's how the serine gets to be by itself. It cleaves the peptide, right? Trypsin cleaves the peptide right here. And then that means phenylalanine must be right here by default. Okay, so that's an example of how you would do that. That is how you would identify the sequence based on the data from the problem including the fragment data. Okay, so just remember when you do this, when you start these, the orders that these are given to you are just alphabetical. The orders in the sequences have to be determined by knowing what each of the chemical agents do. Basically, this is sort of the summary of what we did, right? How many amino acids are there? Well, there were 8, so it was an octopeptide. Do the results mean certain amino acids have to be in particular spots? Now, I made that general because maybe the cyanogen bromide wasn't first and the trypsin data was given first. Well, that's telling us like where the peptide is fragmented, and so it tells us certain amino acids have to be in certain places. That's what that means. And then sometimes, like happened in this problem, right? The phenylalanine had to be here. And it had to be there because there was no other spaces for it to be if the arginine was next to the serine. So you have to infer positions of amino acids in these problems. Okay, here's another problem. And I want you to try to solve this problem on your own. And the deal is this, that I'm going to solve it after. You're going to pause the video and you should try solving it. I will solve it separately from what you're doing, and then you can look at my solution. There's a very good chance that we're going to do it different ways. Okay, so go ahead and pause the video and try to solve this one. I'll solve it and then talk you guys through it. So I think I did this right, and now I'm going to go through the process of checking it and going over it with you. So let me sort of explain to you what I did, okay? So my first step was to figure out how many amino acids were in here. And you notice there isn't a total hydrolysis, so what I have to do is count up the amino acids in the fragments. And when I do that, I get an octopeptide again. Then what I did is I went ahead and I didn't start with the cyanogen bromide. And the reason is it gives me 3 fragments. And actually, I don't want it to be broken up so many different ways, because when you start with 3 fragments, many times the fragments can be in different orders and you can't tell because of the multiple methionines. It cuts in different positions. So what I did is I went automatically to the end and looked at the trypsin data. And I wrote the notes down, right? Arginine and lysine. So notice in this I have, and then I should have done this. I didn't do it on this one, but I'll go ahead and do it now. What I should have done is I should have underlined the arginine and lysine in this section. So there's none in here, right? And then there's a lysine here. Okay, so that tells me something automatically. Like fragment 2 is actually the first fragment, and fragment 1 is actually the second fragment in the sequence. So I know cysteine and tryptophan have to be at the end, and I know that the rest of it has to be here. And what that allowed me to do is allowed me to fill in the position of lysine. Okay, so that's the information I got from that. The lysine and everything else that's with it is in the first half, and the cysteine and the tryptophan are at the end. So then what I did is I looked at the chymotrypsin data. So I'm working myself backwards so that when I do the methionine and it splits it up more ways, I'll be able to put more things in order. But I'm trying to deal with the big pieces and figure out what the big pieces tell me. So again, I list the amino acids that it's going to cleave on the C side of it, because it's always on the C side for these reagents. And I have tryptophan and tyrosine. So here's the thing. If those had not, those two amino acids have to be at the ends of their fragments, because if they're not at the ends of their fragments, they'll create another fragment. Okay, so I'll say that again. Those amino acids have to be at the ends of their fragments, because if they're not, they create another fragment. So that means the tryptophan here, right? I color coded this, by the way. The tryptophan is in position number 8. That's this guy here. Has to be at the end, because if it was the other way around, I would have a cysteine by itself and a tryptophan by it. Well, would tryptophan be by itself? I would get cysteine by itself for sure. Okay, so that's how I did that. And then by inference, right, that's what this fuchsia color is. Cysteine has to be in position number 7. Now, some of the other things that I know, right, is because of the tryptophan being at the end, I know the tyrosine has to be in position number 4. So I'm going 1, 2, 3, 4 because this fragment is 1, 2, 3, 4. I know tyrosine has to be at the end of that fragment, so it's actually going to cleave it here. And then that's how I get the 2 fragments that are shown here. There's the cysteine, there's the lysine, there's a methyrosine, and a tryptophan. And tryptophan, again, has to be the end, otherwise it would cleave off. And then, anyways. So cysteine, by inference, is in position number 7. And now I can go to the cyanogen bromide. So if I look at the cyanogen bromide, I'm going to underline the methionines in there. I meant to actually circle A and not the 4 there. Methionine is here, and methionine is here. So those have to be at the ends of their fragments. There's 2 of them, and that's why I have 3 fragments. So I can guess as to the position of each one, because if I look at the methionine, tyrosine, and valine, methionine and tyrosine and valine are right here. And that gives me the sequence that I get for methionine and lysine needing to be next to each other and needing next to be to cysteine. That means this fragment number 3, is actually the first fragment in the sequence. So that allows me to place the leucine and the methionine. All right, and this methionine I could have got by inference as well, because I should have a valine tyrosine methionine fragment, and that's this valine tyrosine methionine fragment. But because the methionine is in position number 2, the leucine has to be in position number 1. So that's the sequence that I get for that peptide. Okay, so you should be ready, to take on a problem like this, and to check yourself. You could try doing this one down here. Okay, I think this might be a nonapeptide. You'd have to count them up to be sure, and then try to figure out what the sequence is. And I gave you plenty of data to do it with, actually. Okay, again, if you have any questions, if you want to go over that sequence at the end, feel free to contact me anytime.